This recording is
Okay, finally. So, welcome to chapter 9. So this is muscle physiology and a little bit of histology. So we'll kind of build on what we talked about last time when we started the histology conversation with muscle, and we'll get into muscle physiology. So this is going to be focused not so much on the anatomy of muscles. That's going to be lab tonight, next week, for the week after that. This one's going to be mostly focused on how muscles work. Okay, so that's going to be that particular chapter. Just like with the skeleton chapter, 6 was the physiology chapter, 7 was the anatomy chapter. The same thing's true for this one. So this is the physiology chapter. The next chapter is the kind of the muscle anatomy chapter. And I will do the same thing as well on Connect. So for like the anatomy chapter is post like that Connect assignment up there. So you can kind of use it as a review and an anatomy rundown on muscles as you guys are preparing for muscle anatomy. So basically we're going to be taking a look at how muscles function and how they basically work. And so we're going to start off first of all looking at structural elements of the muscle itself, the organization of the muscle, the organization of the cellular level of the muscle, which is a muscle fiber. And then we're going to start looking at some of the more physiological dynamics of like muscle twitch dynamics, relaxation, contraction, excitation, things like that, and actually go down and flesh out how all of this works. Okay. Whoa. Okay. Too big. There, we'll keep it at that. Stop playing with it. Okay, so first of all, we're going to be taking a look at the different types of muscle and our muscle tissue. Of course, we learned about this in the histology chapter, right? So we have 3 main types of muscle. We have our histology, we have our skeletal muscle, which is by far the one that we typically think about the most. Then we have our smooth muscle and of course our cardiac muscle when we take a look at each of these. Skeletal muscle, just to kind of build on what we started off with in just a very, very brief touch. This is going to be the one that you typically are associated with locomotion and movement. So your facial expressions, anything that's attached to your skeleton that requires you to be able to move is going to be a skeletal muscle. For instance, your postural muscles, even though they're constantly contracting, are skeletal muscles, respiratory muscles, respiratory muscles that are associated with like the costal muscles, things like that, are skeletal muscles. So basically the name of the game here is body movement. That's what you're doing here, right? You're moving your skeleton around. Skeletal muscles are under voluntary control and they're controlled by your nervous system, which you would imagine then that the next stop after the muscle physiology one, once we wrap that one up, will be the neurophysiology chapter, right? So now that we kind of have that sort of out there. Smooth muscle, this one is going to be a little bit different, right? So it's going to be largely focused on your organs, your blood vessels, your eyes are smooth muscle, glands, skins, things like that. These are basically automatic sorts of things. So they're typically involuntary, and they're oftentimes sort of working in the background as part of your controlled autonomic responses. Autonomic basically are automatic. So they're on like autopilot. So these are the kinds of muscles that are associated with unconscious movement of your body. For instance, as the food moves through your intestines, as it basically moves from one point in your intestines to the other, it's smooth muscle that's doing that. As your blood vessels are dilating and constricting to regulate your blood pressure, it's smooth muscle that's doing that. So typically these are the ones that are associated with your visceral core. And when you get an AMP2, most of what you're going to be working with outside of the cardiovascular system is going to be smooth muscle. And so the third one, of course, is cardiac muscle. This is only found in the heart. And it's also involuntary and automatic and can be controlled by both endocrine and also nervous inputs. So typically skeletal muscle is the only one that's regulated by purely the nervous system, whereas the other two are also controlled by the endocrine system. Notice one thing, there's a commonality here. When you need some sort of an action that's sort of on autopilot, oftentimes your endocrine system is involved. So your endocrine system is sort of like the Wizard of Oz behind the curtain, right? Pulling all the levers that you're not even aware are being pulled, making sure that everything is running smoothly, sort of in the background, okay? And so that's pretty common to see those autonomic types of systems being driven by the endocrine system. So just to kind of refresh your memory, this is basically what we have in terms of the three different muscle types. So skeletal muscle, you can see, is attached to your bones, allowing your bones to move. And by far, this is what we're mostly going to be focusing on in lab when we are working with the cats and with the models. Is we're focusing on the skeletal muscles that's attached to different types of bones and things like that. We'll talk about different classifications of that here in just a little bit. But if you recall, this is going to be the one that is that long cylindrical type of muscle fiber. And oftentimes we'll have multiple nuclei in a given cell. These smooth muscles, which typically are lining the smooth or hollow organs, are going to be like the gummy worms, basically piled up on top of each other. And you can see how they look very spindly in nature. And they look like, like little gummy worms that are just kind of all sort of squirming around next to each other and kind of pushing each other out of the way. And then the cardiac muscle is going to be this interconnected network of muscle fibers. You can see how the actual muscle fibers themselves are interconnected between each other, and they're bounded by the intercalated discs, which are essentially these gap junctions that allows for simultaneous uniform communication from one cardiac muscle cell to another. And so these little dark lines that you see right here, those are those intercalated discs. Kind of hard to see on the projection, but if I blow it up a little bit, so you can see those like little lines right there. Those are the intercalated discs. Okay. That's the boundary between one cell and another. So this is just kind of like a brief, just a table of exactly what we just mentioned. Some of the stats, if you will, the vital stats of the different cells. Of course, the ones I like to pick out are the cylindrical nature of the skeletal muscle as opposed to the spindle-shaped shape of the smooth muscle cells and the branching of the cardiac muscle cells. Oftentimes at first sight, this is what will allow us to be able to diagnose which one is which under a microscope, histologically speaking. Nucleus, the smooth and cardiac are single, and the skeletal is multinucleate. Generally speaking, skeletal muscles don't have super fancy special cell adhesions, but smooth and cardiac do. Oftentimes are gap junctions, and of course the intercalated discs. And with the striations in this particular chapter are going to be important because we are going to learn what those striations are in this chapter. And of course, we're going to be focusing mostly on skeletal muscle, which are heavily striped. They look like little candy canes, right? So they're all nicely striped. Smooth does not have the striping, and Cardiac does, although it's to a lesser degree. So in terms of its control, skeletal is mostly voluntary, although there are involuntary skeletal muscle. That's the reflexes, basically. So the reflexes are involuntary, but by far they're mostly voluntary. Smooth and cardiac are involuntary because these are the ones that are responsible for managing those core visceral types of movements that you don't need to be bothering yourself with thinking about all the time, right? And sending a message to all the time. So generally speaking, spontaneous contraction is only the domain of your cardiac and your smooth. Not all smooth, some smooth will spontaneously contract, but mostly it's a cardiac muscle thing. Skeletal muscles don't spontaneously contract. They have to be told to contract, which is the reason why we hook up the nervous system to skeletal muscles so that our nervous system can tell those skeletal muscles when to contract, how much to contract, and so forth. So that's the controlling piece of it. So we kind of went through the functions already, I think, in a couple of different ways. Now let's go ahead and take a look at the muscular system overall in terms of its functionality, kind of the same way we did for the skeletal system, right? So obviously, number one at the top is the role of the skeletal muscles, which is movement of your body. That's number one. Maintenance of posture, those are your postural muscles, so they'll basically keep you upright. If you're wondering what is keeping you upright, it's not, you're not upright because that's the way that your body wants to go. It's your muscles that are keeping you upright. And so a lot of times we don't even realize that because it's just, they're constantly contracting all the time. Respiration, your muscles are in charge of that. So if you didn't have musculature, you would not be able to breathe. Body heat, these are the primary heat generators of your body. Which is one of the reasons why when your core body temperature starts to drop, you start shivering, is because your body is trying to generate extra heat to increase your core body temperature. They're typically going to be associated with communication, of course, movement of the facial muscles and things of that nature. Organs and vessels, they're basically in charge of that. The smooth muscle, for instance, is going to be in charge of your organs and your blood vessels. And of course, cardiac muscle is in charge of the contraction of your heart, which is necessary in order to keep all the rest of your body alive. So basically, in more ways than one, if you didn't have musculature, you literally wouldn't be alive. Right? So muscles are pretty important because they basically touch on just about every major system that you can go over. Okay, let's take a look at what some of the properties then are of muscular tissue. Number one is probably the one that we associate with muscle primarily, right? We actually even talked about it in the tissues chapter, that muscle tissue is the contractile tissue. And so contractility, that is to say the ability of the muscle to shorten and to create force by that shortening is what contractility is. And so it can shorten and create a force. We'll talk more about that. It's also excitable. So that is to say that the muscle can respond to stimulus, oftentimes coming from nerves. And so you're excitable and contractile. So not only can you shorten and produce force, but you can shorten and produce force on demand when the nervous system tells you to do so. It's not good enough to be contractile. You also have to be extensile, right? I don't know if that's a word, but it is now. Right, so extensibility, this is basically the ability for your muscle not just to contract, but to also stretch, usually beyond its normal kind of resting length, and this will allow you to be able to contract. Basically speaking, when you're looking at musculature, the more you relax, the more you contract. The more you contract, the more force you can exert. So stretching out your muscles is a good thing. It's priming your muscles to produce maximum force. And then, of course, elasticity, your muscles have to be elastic in nature, which basically means that they can recoil back to their original shape or their original length after they've been stretched out. Think of it as sort of like a rubber band, right? A rubber band has a particular length to it. if you stretch that rubber band out, that's its extensibility, if you will, right? Its ability to sort of go beyond its original resting stretch. But then when you let that rubber band go, what does it do? It goes right back to its original shape, right? That's its elasticity. So it has the ability to recoil. But a rubber band has limits, doesn't it? You can stretch it beyond its ability to recoil, like a slinky. I mean, who doesn't love a good slinky, right? Right, a slinky, we love this thing, right? Because basically we can stretch it out and it's got this extensibility to it, right? And as long as we don't overdo it, it'll come right back to its original shape, right? That's its elasticity. But we all know, you've been a little too rough with your slinky, that if you pull that thing apart too much, you're going to exceed its ability to come back, and then it's all stretched out and distorted, right? Now you got a useless slinky. And so the same thing's true for your muscles. You have a certain amount of extensibility and elasticity so that your muscles can stretch out and then come back to its original shape, but it does have boundaries to it, and you can't overdo it. We'll talk more about that later on. But before we get there, let's cover a little bit of the structure of some of the muscles. The first thing we're going to be taking a look at is the coverings. So when we take a look at the whole muscle, we'll notice, first of all, that there's layers to its coverings. The first one, which is the outermost layer, is called the epimysium or the epimysium. This is basically a connective tissue area that surrounds the entirety of the muscle. And it typically is the outer layer. So this is a layer that merges with the fascia, the muscular fascia, that connects different muscle bundles. So one of the things you're going to notice in cats is there's fascia everywhere. And basically what fascia is it's essentially a sheet of membranous connective tissue. That bundles structures, in this case, muscle, together. So your muscles, your veins, your arteries, things like that, they're not just basically sort of rattling around freely inside your body, just kind of bumping around and just sort of boggling around. They're bundled up, they're wrapped up like a mummy in this fascia, keeping them in place. So that when you're moving your body, they're not just rattling around in there randomly. They're actually bundled together snugly, kind of like being all snuggled into a really nice, cozy, tight sleeping bag. And that's what this fascia is. And so it's a lot of this kind of membranous connective tissue that's in between muscles that bundles them together. And then also this is continuous with that subcutaneous layer of the skin and will actually merge with that underlayment of the skin and kind of fold into that piece. It also is what connects to this epimysium. So your muscle cells, very similar to the way we talked about the periostea, being able to weave into the tendons and connect the tendons by basically weaving those similar connective tissues together. This is the same idea. Here you've got a connective tissue outer layer, the epimysium, which is basically weaving itself into this fascia sheet and then tethering itself to that hard, solid structure that will basically keep it stable and keep it from moving around too much. So as we continue to go inward, we're going to get to the perimysium. So epi means upon, the mysium, the mysium, which is the musculature. Peri means around, the mysium or the mysium, which is the musculature. This is basically a loose type of connective tissue. Now notice when you're looking at the epimysium, the connective tissue here is typically dense in nature and collagenous. But in the perimysium, you're talking more about loose connective tissue. And what it's going to do is it's going to surround groups of muscle fibers. And these muscle fibers, these groups, these bundles, if you will, are what's called fascicles. And so this was basically where you're going to get your blood vessels passing through there, and your nerves are going to be moving in between these vesicular bundles of muscle fibers, communicating with each of these different muscle bundles, whether they need to contract or not. And then the endomysium or the endomysium is also loose connective tissue. So endo means inside or into the musculature. This is basically going to be separating the individual muscle fibers that are within each fascicle. So if you're keeping track, we've got 3 levels. Matter of fact, in a lot of ways, the way your muscle is built is like the cable of a suspension bridge. Has anybody ever seen a cross section of the cable of the suspension bridge, whether it's, I think, which is the New York suspension bridge? Is that Brooklyn Bridge? This is a suspension bridge. Golden Gate's a suspension bridge. If you take a look at them, those big gigantic cables are actually not just one cable. They're a cable of cables of cables of cables. So if you take a look at one of those big things in cross section, you'll notice that it's got like smaller cables that make up the gigantic cable. And then within these are smaller cables, which make up those. And then inside these are smaller cables and so forth as you get smaller and smaller and smaller. So it's like a cable of cables of cables in different levels. And that's kind of what a suspension bridge cable looks like. That's exactly how your muscles are built. So at the very base of it are the muscle fibers, which will then cluster together in a bundle of cables to form the fascicle. And then the fascicles will bundle together in multiple fascicles to form the muscle itself. Okay. So then the collagen from these connective tissue layers will essentially be part and the same of the collagen that we see in tendons. So you can see how kind of everything is fitting together nicely, like a nice little jigsaw puzzle, right? Because you've got this collagenous connective tissue in your epimysium, which is interweaving and kind of an extension that's coming out to form the tendons, which interweave with the periosteum as you connect your muscle to the bone. And then that tendinous piece is essentially an extension of the epimysium. So you can see how the connective tissue layers are all kind of integrating with each other. And in some cases, instead of connecting to the bone, you're interweaving with the muscular fascia. Or another type of tendon called an aponeurosis, which is essentially a large sheet of a tendon. A good example is your epicranius, which you have 2 bodies. You'll see 2 naming conventions for epicranius. It's the muscles that basically cover over your head. Some texts will call it the epicranius with a frontal belly, that's the frontalis here, and an occipital belly with an aponeurosis. Basically, it's a sheet of tendinous membrane, connective tissue membrane that connects those 2. Other texts will consider this a separate muscle in its own right, the frontalis, and then the occipitalis back here. And then they're connected with this aponeurosis. But this aponeurosis is literally just like a little sheet of connective tissue that's over the top of your skull. And so that will be your attachment. So you can see a pattern here, can't you? It's the same pattern over and over and over again. You have a connective tissue outer layer. And then you have a middle layer that is kind of like up to you, right? And then you have an inner layer, which is oftentimes very physiological in nature. And so you see that pattern repeating itself over and over and over again. You even see it when you get into AMP2, when you start looking at the structure of the intestinal wall or the structure of blood vessels. So you see this pattern all the time. Okay, so then once we actually now have the structure of the liners, what about the vasculature and the nervous integration? So the nerves that are basically feeding the skeletal system or telling the skeletal muscles to contract are what's referred to as motor neurons, right? So basically, they're communicating movement. They're telling your skeletal muscles to contract. That's what they're doing. Generally speaking, a single motor neuron is going to be in control of several muscle fibers. So it's not just talking to one muscle fiber at a time. It's going to be several of those muscle fibers. So each muscle fiber is basically connected to a particular component or piece of the motor neuron, which is called a teledendrion. We'll talk about that later when we get to neurophysiology. Generally speaking, you'll have an artery and a couple of veins kind of wheedling their way through there. And then the nerve, which will be kind of going through these connective tissue layers. And then you'll cover the muscle fibers in these capillary beds. And this is basically where you get your gas exchange. And so that's going to feed your muscle cells with the necessary nutrients and energy that is required in order to be able to contract, right? So it's a very vascular area. So this is kind of what it looks like. So here you can see your epimysium, which is this outermost portion covering the entirety of the muscle. And so that's your first and your connective tissue surrounding area. And then generally speaking, this is going to be connected to the muscular fascia, and the fascia, which bundles up the muscle, will oftentimes sort of peel off in like little sheets and will cover oftentimes other muscles as well. So you'll see the fascia kind of bundling up groups of muscles within them. And so here's your epimysium, which is covering the entirety of the muscle. And then as you start to get down to the size of the fascicle, so for instance, this right here is going to be one fascicle. This is going to be surrounded by the perimysium. Now notice this is where your blood vessels and your nerves are going to be attaching to the muscle fibers, which themselves are inside the fascicles. So when you take a look at each individual little circle inside the fascicle, this is going to be one fiber. And then around that, you're going to see this little blue kind of halo around those individual muscle fibers, which are right here. That's going to be your endomysium. This is the one that's surrounding those muscle fibers. So here you can see a cutaway or a cross section of it here. So you can see the epimysium is going to be basically up here. This area right here is the epimysium. The perimysium, so here's one fascicle. I'm going to surround it right there. So there's your fascicle right there. That's going to be the perimysium that I just highlighted. Here's another fascicle over here going off in that direction. So they're kind of right next to each other. And then the dark eyeliner around each of these little individual circles, each of those individual circles is a fiber. And each of those little dark eyeliner areas around each of those fibers is the endomysium. So that's the layer around them. And here's another high magnification view of that as well. So you can see then that basically the fascicles are essentially wrapped in the vasculature. So you can see the arterial and the venous and also the nervous inputs associated with these. Okay, so let's kind of start working our way through these one step at a time, because structurally, I'm talking about muscle structure right now, they're structured very differently, right? So we kind of ran through that, and we got a good view of what those structural differences look like. But let's take a look at the skeletal system first, or the skeletal muscle first, and talk a little bit about where they came from and how they came to be. So basically, skeletal muscle is a development from an early progenitor of muscle cells called myoblasts. So myo refers to muscle, and a blast cell, where these are these developmental cells. So they're typically responsible for creating new tissue or becoming new tissue. So these myoblasts are embryonic cells. And so what happens is, I think, remember I mentioned before that there's two ways to get multinucleated cells. One is to replicate without division, right? So normally when you go through cell division, you learned about this in general bio, right? So you usually replicate and then you separate and you become two cells. If you replicate without separation, then what you've got is two nuclei in the same cell, right? So that's one way to become multinucleate. The other way is to do this, is to basically take two cells, two myoblasts in this case, each of which has their own nucleus, and for these guys to fuse together. So if they fuse together, then they become basically a single cell with two nuclei in it. And if they fuse another one together with it, then they become a threesome with three nuclei in it, even though what happens is they become this just really long, elongated cell with multiple nuclei. And that's exactly what we see with skeletal muscle. So you can see how you have multiple nuclei in them because of the fusion of multiple myoblasts, basically creating these long cylindrical cells. So skeletal muscles can be quite long, right? So talking about 1 to 4 millimeters long, this is basically visible. So you can basically view this with the naked eye. A millimeter is 1/10 of a centimeter. So it's really tiny, but you can see it. They can also get pretty long. They can get up to 1 foot long, depending on where you are in the body. The diameter is considerably smaller, of course. So generally you're between 10 to 100 micrometers in diameter. So you typically tend to be fairly small in diameter. Although if you think about it, that's actually a pretty good range, right? That's a 10-fold variation in range. That's a pretty huge range for something like that. Okay. Their striations, which we'll talk about, is actually part of their function. And so normally when you take a look at the number of muscle fibers you have, the number of muscle fibers you have pretty much stays the same. So when you're building muscle, you're not adding more muscle fibers. What you're doing is you're taking the muscle fibers you have and you're making them bigger, right? That's what hypertrophy is. Hyper, big, more than, trophy is basically structure, kind of structure, bigger than the original structure of muscle fibers. So you're basically adding more stuff in the already existing muscle fibers that you have. You're not adding new muscle fibers to your body. So when you take a look then at some of the histological markers of your muscle fibers, this is an important list to kind of go through the parts of your muscle fiber. Now, a couple of things to really sort of dissect and to make sure we understand terminology-wise is in muscleology, muscle fiber is basically synonymous with the cell. So to say muscle cell and muscle fiber is exactly the same thing. That's what a muscle fiber is. It's the cell of the muscle. Now, the reason why I mention this is because sometimes from general bio, many of you rather enjoyed perhaps the cell chapter when you talked about all those organelles and things like that. Some of you maybe didn't. But either way, because this is a muscle cell, just like any other cell, that basically means that it has all the working parts of any other cell, including all the organelles that you learned about in general bio, right? Like mitochondria and smooth endoplasmic reticulum and stuff like that, right? So it's got all the normal working parts of a cell. We just happen to call it muscle fiber because, well, muscle people like to be special, right? But it's the same thing. Sarcolemma, which is another term that we run into in muscleology, is basically, to decode that, is simply the plasma membrane of the muscle cell. That's what that is, okay? Just in muscleology, we call it the sarcolemma. And then within the cell, instead of saying cytoplasm, which is actually what it is, we call it the sarcoplasm. Okay, now wait, I already know what you guys are thinking. You guys are probably thinking, wait a minute, wait. If this is just basically the same thing, smooth, cytoplasm, plasma membrane, cell, then how come we had to go through the torment of creating a second set of terms? Is it just because you want to make my life more difficult in anatomy and physiology? Let's face it. Sometimes when you're studying A&P, it kind of feels like somebody's coming for you, right? It's like, you did this on purpose. You created all these names and terms just to make my life miserable. Well, possibly, but I never put it past med school to intentionally try to make your life more miserable than it ought to be. That's kind of med school shtick. That's kind of what they do, right? But no, what this came from in a lot of these terms are because the cell people who are studying the cells were studying the cells separately from the A&P people. They didn't realize they were studying the same thing until much later. And so as a result, the A&P people came up with their own set of terms, not realizing that they were essentially looking at the exact same structures that the cell people were looking at. And so what looked to them to be like a very novel structure, this muscle fiber, was actually just a basic, common, everyday, ordinary cell. Okay, that's the reason why these two sets of terms came to be. There are some unique structures, however, to the muscle fiber that most cells do not have. And one of those is in number 3, your transverse tubules, or the T-tubules. These are basically inward invaginations of the sarcolemma, and they dive deep like a mine shaft into the center of the cell. So if you were to take a look at the sarcolemma of a muscle, the T-tubules will basically just dive deep into the interior of the cell. There'll be a deep invagination like that. And that basically gives you access to the internal portion of the cell itself. Normal cells don't do that. This is something that's special about muscle cells, right? Most other cells, they'll have an entire signal transduction communication strategy where you use receptors and signaling molecules to communicate to the nucleus what to do, things like that. That's the way they do it. But these guys prefer to have this dip down deep into the cell membrane, which is unusual. But when we take a look at why it's there, it'll make sense why we do this in muscle cells as opposed to another type of structure. The other one that they also have is what's called the sarcoplasmic reticulum. This is basically your smooth endoplasmic reticulum. It's a little modified, but for the most part, it's smooth endoplasmic reticulum, just like you see in any other normal cell. Its job, however, is to store calcium. And so at the tips or the terminus of these smooth and this sarcoplasmic reticulum are swelling areas, enlarged regions, kind of swollen areas called terminal cisternae. And these guys will lay adjacent to the T-tubules. We're going to see a picture of this here in just a second. Generally speaking, you have 2 terminal cisternae that basically flank a T-tubule. And we call this structure a triad. So basically what's going to happen is along this little T-tubule, you'll have your smooth endoplasmic reticulum with these terminal cisterna basically kind of flanking the edges. And then this terminal cisterna will kind of go off and do its own little thing. And so forth. So it'll kind of like, that's a really bad drawing, but I can't really draw. So essentially what will happen is they'll kind of have these like little areas like that. And you'll see that this region right here, the 2 terminal cisterna with your triad or with your T-tubule is called the triad. So the reason why you have the T-tubule dipping down in there is to flank these terminal cisterna. And there's a very good reason for that, because it's part of your muscle contraction mechanism, which we'll talk about a little later in the chapter. This is kind of what it looks like. This is your muscle fiber. So typically you're going to have your nucleus or your multiple nuclei, your sarcolemma, which is covering the cell itself. That's the plasma membrane. Notice inside you're going to have mitochondria, which you need in order to make energy, right? So you need that ATP in order to contract your muscles. The sarcolemma, however, will dip down. So here you can see your T-tubule basically dipping down and vaginating and creating this long, deep shaft-like structure. And it doesn't just form a lateral, it actually kind of curves around in a circle. So it actually kind of circles around the backside of this large cylinder-like structure called a myofibril. And it'll basically kind of enter and go back up. So essentially kind of in circles or in wreaths, these two pieces are of myofibril. And then you can see here how your T-tubule is flanked by the terminal cisterna here. Cisterna, and T-tubule, CTC. So this basically forms ultimately together your triad. So terminal cisterna, T-tubule, terminal cisterna forms your triad. So your triad is basically formed by this structure right here inside the box. So your sarcoplasmic reticulum is designed to flank that T-tubule. Good. And here you can kind of see how the T-tubule actually wraps around that little myofibril cylinder, which is pretty much a long cylinder of protein. And we'll take a closer look at that here in just a little bit. But these guys are your myofibrils, which are very important to contraction. So that's basically your muscle fiber. Notice it's a cell, just like any other cell. It's a highly modified cell, no doubt, right? But it's still a cell. Now let's go ahead and take a look at these myofibrils and some of the proteins that are in there, right? Because these are basically what's going to create these mechanical or physical components of the actual muscles, and it underpins the mechanism of their contraction. This is the important piece. The myofibrils are really, really important because these guys are essentially protein filaments that will form these long cylindrical shapes. And generally, these long cylindrical shapes will be the myofilaments. So they'll form long fibers. It's long fibers of filamentous protein. Gives you myofilaments, which makes up the myofibrils, which are bundles of myofilaments. And these are the ones that cause contraction. So notice we've added a couple more layers to our cable of cables, right? Starting from the bottom up, myofilaments will bundle together to form a myofibril. Myofibrils will bundle together within a muscle fiber. Muscle fibers will bundle together to form a fascicle. Fascicles will bundle together to form the muscle. That makes sense? So starting from the bottom, going all the way up. Okay, let's take a look at these myofilaments. So you have two different types of myofilaments. The first one is the so-called thin filament, which is composed of a protein called actin. Does that one sound familiar? Do you remember that from your repressed memories of general bio? Remember the cell structure chapter? At the very end of it, we talked about the cytoskeleton. We talked about things like intermediate filaments. We talked about microtubules. And the third one, microfilaments, which were made out of actin, right? So actin is actually the main component, actually the only component of the main component of the thin filaments, okay? Now there's another filament called the thick filament, which is made out of myosin. And myosin, when you take a look at it, is a long shaft-like piece, but this is essentially a motor protein. Did you guys talk about motor proteins in general bio? We talked about flagellar movement and the way the dynein walks along the flagella, the microtubules. Sometimes some professors do, some professors don't. In any event, when we start talking about muscle contraction, I'll do an intro on motor proteins just to make sure everybody, if you've forgotten about it, that it kind of refreshes your memory. Okay. So then these myofilaments will then be organized into the most important subunit of the muscle, which is the sarcomere. The sarcomere is essentially the contractile unit of the muscle. What you do to the sarcomere, you're going to do to the muscle. So, yeah, I think I got it all. So that's kind of what it looks like, taking it from the bottom up. So you have your myofilaments, which you have your actin filaments and your myosin filaments, which will then form the myofibril. So you're going from smaller to larger. And then within the muscle fiber, you have your myofibrils making up your muscle fiber. And then your muscle fiber, which is going to be surrounded by the endomysium, is going to be bundled up into your fascicles. So here's a fascicle here, here's one here, here's one here. And your fascicles are going to be bundled up in your muscle. So you can see how you can kind of break it down. This is, by the way, if you're wondering, is kind of what we were looking at the very beginning when we looked at the lecture outline. Didn't make a lot of sense then, but hopefully it should make a little bit more sense now. Maybe, right? Or it will make a little bit more sense with a little bit of study as you guys get into it. Now, here's the reason why we cover this structure. Because when you're talking about the function of a muscle, especially its contraction, the one thing you have to understand is that this is one of those situations where the theme of structure sets up function is absolutely critical. So when you have a function to create a load, to shorten and create a force, then the mechanism to shorten requires these structures in order to shorten. So the reason why you have your muscle structured the way it's structured is so that it can do its basic function of shortening and creating a force. Okay, so structure is very tightly aligned with function when it comes to skeletal muscle. So the sarcomere itself is going to be your basic functional unit of the muscle fiber, and it's broken up into different pieces, things that will basically, they'll be a little abstract at first, but with a little bit of study, they'll start to make a little bit more sense because it's very logical the way the sarcomere is organized. The first place we're going to start is with what's called the Z discs. The Z discs are basically a filamentous network of protein, and this is going to be the attachment point for the myofilaments. It also represents the wall or boundary of a sarcomere. So from Z disc to Z disc is one sarcomere. So that defines where your sarcomeres are. Now, the regions of the sarcomere are going to be referred to as mostly bands and zones. So within this, you're going to have what's called the I band, which is an important band. This is a light staining region of your sarcomere. It basically has a Z disc in the middle of it. But basically, this is the area where it'll extend to the ends of your myosin filaments. Now, the important thing to understand here is that in sarcomeres, myosin stains darkly and actin less dark. So this is going from myosin end to myosin end. That's the reason why it's lighter staining, because essentially what this is a region of actin. Okay. The A bands, this is going to be a central region, a dark staining region. This is your region of overlap between actin and myosin. So you basically get the staining darkness of actin along with the staining darkness of myosin. So the two of them overlap, giving you an extra dark look which is the reason why the A bands are typically dark. With the exception of the center, there's like a little white stripe in the center. And that white stripe is what's called the H zone. The H zone is in the A band. This is basically where the actin and the myosin do not overlap. That's usually in the middle of that A band. And then in the middle of that white stripe is a little thin, dark line. It's right in the middle of the H zone. And so this is basically a thin, darker line. And this is just kind of like a very little thin line of filaments, very delicate filaments that's holding the myosin in place. So this is basically where your myosin is tethered to. So it's anchorage point. By the way, this alternating light, dark, light, dark is what creates that light and dark striation pattern that we see associated with skeletal muscle. So the striations that you're seeing in skeletal muscle, you're seeing the boundaries of the sarcomere, the multiple sarcomeres that are lined up on each other end to end. Okay. And so those are like the major areas of the sarcomere. So essentially sorry, I just said that. But in addition, so they're lined up basically to create the striations, essentially what that first bullet is. In addition to this, there's another protein in there called titin. So titin filaments are these elastic chains of amino acids that kind of make that extensible elastic character of kind of like a bungee cord. Of muscles. Right? Extensibility, stretching beyond your means, elasticity, coming back to your original shape. And so this is kind of what the sarcomere looks like. This is definitely one of those situations where a picture is truly worth 1,000 words. So there's a couple things going on here. And I think I might try to see if I can, I tried this before. I feel like I failed miserably because it doesn't really do anything for us because the smallness of the screen is not PowerPoint, it is Zoom. Yeah. A little bit better. So here's our sarcomere. So first of all, let's take a look at this little zigzag line that you're seeing here. I'm going to try not to write on this too much because it's really poofy, right? But this little zigzag line right there, that's your Z disc, and here's your other Z disc right there, right? So from Z to Z is one sarcomere. Then the I bands, remember these are the light staining bands, are basically with a Z disc in the middle, this region of light stain. So here you can see this lighter area of the micrograph where you can see that I band. The dark line here is going to be your Z disc, okay? And so here's your other I band over here. Now in the middle of the sarcomere, you're going to see this dark region here, like that. This is going to be your A band. This is that region of overlap. And right in the middle, you'll notice is this H zone with this little M line going right down the middle of it. So it's a little darkened area going right down the middle of that H zone. And so if you take a look at this, this is basically what it looks like. So here is one Z disc over here. Here's another Z disc over here. typically attached to your Z discs are these little actin chains. That's these little purple chains right here. And so they're basically kind of laterally moving across. In between and tethered to this M line are going to be the myosin filaments. And the myosin, you can see, is basically attached to the actin. So if you take a look at this and kind of blow it up a little bit, you'll see that you've got this region of overlap right here. That's your A band right there. The region where there is no more myosin, it's just actin, is going to be your I band. And then tethered to the Z disc, you're going to see this kind of coil of protein here, which is titin. Titin has the effect of basically locking on to the tip of myosin and basically kind of goes across and then to the other side. But basically what it does is stabilizes myosin. It gives myosin something to attach to kind of pull all this together, because you don't want any of these parts kind of moving around just randomly. So when you take a look at this in cross section, you're just going to see a couple of things. If you cut it within the I band, what you're going to see looking end on is you're looking basically down the center of the sarcomere. You're going to see these little purple actin spots. But here's the problem with this particular cut. It's not quite accurate, is it? Because you're also going to see in between this titin. And they don't show it in this one, but titin is in there, right? So you'll see those little titin filaments. If you cut it in the region of overlap in the A band, what you're going to see is you're going to see your actin filaments surrounded on all three sides by your myosin. So your myosin basically has a 360-degree orientation of actin all the way around it. It's like an octopus, basically connected to an actin filament all the way around itself. If you take a cross section of the H band, this is the area where you have myosin, but you've exceeded the length of your actin. So it's a myosin-only area. You'll notice you're lacking actin. So there's no actin now around this, just your myosin in this particular case. If you take a cut between the M line, you're going to see that you have this kind of filamentous structural protein network that connects these little myosin pieces. So these little green circles are the rod portion of the myosin. And it's interconnected by this scaffold-like structure. That's the M line that kind of connects them all together. So it's very interesting when you kind of take a look at it that way, how these all sort of fit in these particular bands and zones associated with the sarcomere itself. Now, when you take a look at the myosin molecule, or the myosin molecule itself is basically a motor protein. And what that basically means is it's a single myosin is essentially a single shaft of a protein with a golf club-like head. And the head will basically pivot back and forth under the stimulation of ATP. So what's going to happen is if my arm is a myosin and my hand is the head, it's going to grab onto something like an actin filament, and then it's going to pivot and pull that actin. And so the more heads I have, like I've had a whole bunch of wrists on my arm, that'd be weird, right? But it is Halloween, so anything's possible, right? So you can grab onto this with multiple hands, and you can just basically keep pulling it and pulling it and pulling it, just like you're pulling a tug-of-war rope hand over hand. And that's basically how myosin will attach. So it attaches all these little myosin heads to your actin filament, and it'll basically pull that actin like a tug-of-war rope, and that's what causes it to shorten. Remember, the actin itself is tethered, it's anchored to the Z disc. So if the myosin is doing the tug-of-war thing and pulling the actin along, then the actin, which is tethered to the Z disc on both sides, is going to start to shorten. And basically, these guys are going to start to pull inward toward each other, shortening that distance. And so you'll get a shorter sarcomere because of that. As long as you have a region of overlap. So as long as your myosin heads have actin to connect to, then you'll be able to pull those two actins together. So from either side, it's like I'm pulling on one actin over here, one actin over here, and I'm slowly pulling them toward each other like this. So I'm kind of using my fingers to kind of pull on those actin chains, and I'm pulling them inward toward each other. As I do that, those Z discs are being pulled by the actin chains closer and closer together, shortening the sarcomere. That's basically how you shorten the sarcomere, and it's also how you contract your muscle. So that right there is, I just kind of gave you a little bit of a cheat ahead, is what we refer to as the sliding filament model. That's sarcomere in short. So let's take a look at the actin filament itself. And we'll probably have maybe just enough time to get through this. So there's more than just actin in your actin filament. The main protein that you're looking at is actin itself, but there's a couple of different types of actin. The first type of actin is what's called fibrous actin, or F-actin. So this is basically, will form a double helix chain of two lines. So basically it'll kind of form this long kind of chain-like structure. So there's one, and then the other chain will wind around it. So this will kind of create this sort of double chain-like structure. That's what filamentous or fibrous actin is. The other one is what's called G-actin. And G-actin is important because on G-actin, you have an active site. And this basically is a site where your myosin heads will bind. Because remember, if we said that the myosin heads attach to the actin, it has to have a binding point, a spot for those myosin heads to make attachment to. And those are what's referred to as these actin-myosin sites. So there's two forms of actin in the actin chain. There's another protein involved in actin as well, tropomyosin. So tropomyosin basically is a very long, kind of a rope-like protein. And basically it'll kind of weave along like a little woven strand in a tri-threaded rope along this groove. And what it'll do is it'll basically fold in that double helix of F-actin. And so it's kind of woven around in there. And then your fourth protein that you have in your actin filament is troponin. And troponin basically has three subunits to it. It does three different things. The first thing is basically has a subunit that will bind to actin. So that's the first subunit. The second subunit's job is to bind to tropomyosin. That's that rope-like protein that sort of threads its way through the F and the G actin. And the third one is it binds to calcium. And so, generally speaking, you have a complex of proteins now that are kind of interspersed between the ends of tropomyosin molecules in the groove between these F-actin strands. And so generally speaking, when you take a look at the tropomyosin troponin complex, what this is going to do is it's going to regulate the interaction or the access to these G-actin sites. So they basically regulate access to the G-actin sites. Why? Because what we didn't mention before is when tropomyosin weaves its way through this actin double helix, what it does is it basically covers the G-actin sites. making them inaccessible to myosin. Which is important because you don't want your muscles to be bound to myosin all the time. Okay, so that leads us to the myosin, but this is a good place to start for next time because we're at our witching hour. We got a pretty good dent in there. We're talking a little bit about structure, and then we'll be able to get a lot of work done for Thursday.